All right. Well, let's get going. Welcome to the inaugural Ecosystems of Learning series. So Regenerative Medicine Manufacturing Society is launching a new pilot series that's free and open to the public to learn more about advances happening in the field of regenerative medicine. Since it's free, we're conducting this pilot series. It has limited enrollment. After the pilot series, we'll poll the audience and determine how best to grow these series if there's continued enthusiasm. We will also be recording this session. Before we start our program, I'd like to make some quick introduction for RMMS. This is a professional society dedicated solely to advancing the field of regenerative medicine through manufacturing. Society members include industry, government, academia, and nonprofit organizations. We're an active group seeking to share our knowledge and work together to create solutions to manufacturing challenges. The ultimate goal is to develop standardized manufacturing processes to facilitate the smooth and quick transition of new therapies to market for the benefit of patients. And our first speaker is Dr. Stephen Bauer. So Dr. Bauer, he is the Chief Regulatory Science Affairs at WFirm. Before joining WFirm, Dr. Bauer had 3 decades of FDA experience in regulatory science, regulatory submission review, policy development, and standard development for regenerative medicine products. Today, we will hear some of his perspectives on the development of standards for regenerative medicine and launching a new center of excellence to assist companies with adoption of standards.
Thanks, Josh, and thanks everybody for attending. So we'll get the slides up in a second, but I will say my colleagues and I at FDA and I were quite involved in helping facilitate standards development. And I'm just going to talk a little bit about, how standards are developed, what they're useful for, that sort of thing today. So it looks like the first slide's up. You can go to the second one, Josh. I guess from the first slide, we should say you'll hear from Carolina Lucchesi from ATTC as well, to talk a little bit more about standards in another challenging area that I won't be covering. So go ahead, Josh. Thanks. So we're all interested in this promise of regenerative medicine because of the idea that we're going to be able to cure, mitigate unmet chronic disease medical needs. And there's so many very novel and promising arenas to do so. But there's also a lot of challenges because many of these things are based on cells and tissues, and there is complex biology about that. I'm going to elaborate on that in just a second. So next slide, please. So I did want to define the goalposts here because standards play a role in potentially helping people get over the goalposts faster. But if you really want to have something successful from the medical treatment therapeutic side point of view, the goalposts are set in FDA laws and regulations. And what you require is to show through analytical and clinical testing that your product is sterile, pure, potent. You know what's in there in terms of identity. It's stable, it's safe, and it's effective. And all those are challenging goals. They're the ones that were developed mostly with small molecule pharmaceuticals in mind, but they are applied. And there is a framework that the FDA has set up for dealing with more complex biological entities like cells and tissues, tissue engineering things, what falls under the rubric of regenerative medicine. Next slide. Oh, next slide, Josh.
Sorry.
So I sort of alluded to the challenges for regenerative medicine, but there is a tremendous biological complexity of cell-based products. There is a challenge to find meaningful characterization schemes that reflect especially potency, efficacy, but also safety. There's challenges around that. There's challenges around the scale of time for manufacturing for some of these things and their stability and shelf life, if you will, for product release. And there's continually advances in manufacturing technology and even places where you manufacture. We're now thinking of manufacturing things in space. But all of this level of complexity is higher than you would encounter with small molecule pharmaceuticals and even protein biologics. Next slide, please. So this is just a scheme around stem cells, but it applies pretty widely if you have a complex manufacturing scheme here relying on self-renewal, so you can expand populations, and then commitment differentiation, terminal differentiation related to the function you want from those cells in the clinic. These things are affected by developmental programs that are built in by nature, but also environmental influences. The question is really, can we develop ways to identify the quality of these things that we measure, the quality attributes that are really predictive of safety and effectiveness? That relates to those ideas I showed earlier, purity, identity, potency. If I could have the next slide. So some of these things, if not all of them, are amenable to standards development. And that's so exciting and an imperative to, help the field, facilitate development in this arena, because standards can do many, many things. They can reduce the cost and they can increase the speed. And I think one of the most cogent things on this particular slide, which I borrowed from the standards coordinating body, is that they can accelerate the regulatory review of therapies that employ these standards. So they also, can help facilitate innovation during product development and so on. So there is a need and desire to implement and develop standards, and the acceleration of regulatory acceptance has accelerated itself. A big recent step has been made where FDA has now set up programs to specifically recognize standards relevant to regenerative medicine. FDA has used standards for years, like people are familiar with USP and so on, but now we're moving into this realm of supporting regenerative medicine. If I could have the next slide. So just a broad overview of standards, because often people aren't aware of what standards means to a regulator or to a NIST person or to, whoever you're talking to. But the types of standards include documentary standards, standard reference materials that actually can be used and validate measurements, and then standard reference data even can be used. And these kinds of standards are all things that could be under development and relevant and important for regenerative medicine. If I could have the next slide. So one question is, how can standards be developed? How can they fit into this arena of regenerative medicine? FDA recently had a webinar that I would recommend to you if you're interested in this topic, because it introduces this newly set up CBER Consensus Standards Recognition Program. This slide has a link to that webinar, and that's available online now. So anybody who's interested to further follow up can look at that webinar. So next slide, please. So the Consensus Recognition Program, there was an announcement last November that they had actually recognized 25 standards. And this was just part of a chart. I'm going to elaborate more on a few of these, but this chart, which is on the CBER website, gives you a recognition number, and then it's called SDO, that's the Standards Development Organization. That is a category of groups of people that get together on a voluntary basis and collaborate together to develop these voluntary consensus standards. That's important because not all standards are made by SDOs. The ones that CBER emphasized are these ones that come from these SDOs. But these are the ones that are currently most relevant to my talk today. And then on this chart, you'll see the title, the year, the recognition status can be complete or partial. In that case, people thought at FDA that the proposed standard had really important elements in it, but not the entire thing was relevant. So that's a partial recognition. And you'll get a standards recognition summary. And that's a link from this chart that tells you if it's, completely recognized or only partially, and what FDA considered in recognition of that standard. Next slide, please. So I wanted to mention a few that I thought were relevant and of potential interest. They're on that chart. One is just the general category of testing and characterization of cell therapy products. Another one is relevant to cryopreservation for those types of products. Then there's one relevant to tissue engineered medical products. There's one relevant to transportation. And then an example of a bioassay, automated colony forming unit assays. And then NGS is a tremendous tool these days, and subject to AI, there's a massively parallel sequencing standard. And then another quantitative bioassay, in vitro osteoblast differentiation assay. So if I get a next slide. So standards can help people with scientific challenges, manufacturing challenges, and standards can lead to improved cell characterization and manufacturing processes. There's more need for standards for analytical techniques, or standards for bioassays, for quality attributes, or advanced manufacturing approaches. And there are standards for shipping and logistics and data standards, and I mentioned a few examples of those, but more could be done and more is needed. If I could have the next slide. I think what we would like to see happen is to have more participation in standards development. I mentioned that these SDOs are voluntary consensus community standards. So it does take some commitment and expertise and work to get these standards through. I was involved in many of those things at FDA, like ISO standards and some ATTC standards. Again, they're widely useful for a variety of different important ways to accelerate progress. And the RegenMed Hub and all our partners is a potential source for participation and helping develop a center of excellence for standards development for advanced manufacturing. And with that, I think I'll pass it over to our next speaker, and thank you for your attention.
Perfect. And so next up, and if you do have any questions, please put those in the chat, and we will have a Q&A session directly following this next speaker's presentation, who is Dr. Caroline Lucchesi. So Dr. Lucchesi is currently the BioNexus Foundation Principal Scientist at ATC's Microphysiological Systems Division, where she is pioneering, leading the efforts on development, qualification, and commercialization of biologicals for MPS to accelerate the adoption of this technology. And today we'll hear about some of the work that she is doing with helping to standardize these microphysiological systems and how it's critical for their eventual adoption by the field. And let me go to share my screen. Back up. And take it away.
Okay. Thank you, Josh. Thank you, Steve. Good afternoon, all. Today, I'll be talking about the Foundation for Standards in Regenerative Medicine, but in the context of microphysiological systems. So I hope that some of you might be interested on it. And we can go to the next slide. So let's start with who we are and what we do. ATTC is a nonprofit organization that was founded in 1925. And in 2007, ATTC became the first biological resource organization to be accredited by the American National Standards Institute as a standard developing organization. And for nearly a century, ATTC has been enabling advances in science that benefits the society and enhances human life by providing biological resources. In the past, ATTC built its collection by cells and microbes by being deposited by research scientists, which continues to grow. And today, our scientists dedicated and focus on developing and producing new products to meet the growing and ever-changing needs of the community. But we also focus on investing in new technologies, which one of them is the microphysiological system. So that's ATTC products and service will remain the definitive standards for the biological research in the future. And we can go next. So why MPS? If we take a look of the traditional methods that we use in drug testing, we are aware that they are often off target and they don't generate human relevant data, posing a big issue to safety efficacy during the clinical trials. And about last year, Biden signed the FDA Modernization Act 2.0, allowing pharmaceutical companies to submit an IND using alternative methods to animal models, and MPS rose as the promise to improve and expedite the overall discovery pipeline by increasing the productivity, minimizing the failures in the safety efficacy, and allows for more drugs to enter the clinical trials, especially if used in combination with other technologies such as computational models. Also, since it's an in vitro system, it enables non-clinical study of a subset of the population and is often excluded from clinical trials, such as kids, elderly, pregnant women, and people with pre-existing conditions, improving then the overall diversity that are so much in need in the clinical trials. And we can go next. And with the need for changes, a lot of attention was placed in the MPS and multiple technologies start to emerge. Each one of them has its capacity and capability depending on the primary organ that it was focused when the technology was developing. So people try not to compete at that time. So it was working on heart, for example, had a different feature than platforms that was specializing in liver. And here we have a table of multiple features, which makes the life of the end users very difficult when selecting a platform. So things to keep in mind is what it is the organ or function you are trying to mimic and what is the question you are trying to answer. So I'm not going through each one of these attributes, but I'm just giving you a flavor of what it should be considering. And to date, there is no guidelines regarding the technology engineering, such as the membrane, material, throughput, stimulus, need of flow or not, and others. And even though there are groups that recognize the importance and started to work with multiple stakeholders on guidelines. So it's important when using MPS to encourage to report a detailed protocol regarding fabrication, the technology features, troubleshooting devices, and the data, which is also a criteria in many journals for publication. And we can go next. So independent on the platform of choice, the cells are a very important requirement. While the primary cells are a good option, since it's the closest to the in vivo physiology and allows higher predictability, some of the cell types are difficult to obtain due to the methods of isolation, difficulties in preservation, or even organ availability. So they also have a limited lifespan, and in consequence, there is a constant need to be assessing new donors. Cell lines are more reproducible and easier to culture, but they might differ from the tissue of orange, which results in not be fully functional. iPSCs have a great proliferative capacity and have the advantage of differentiation in multiple tissues coming from the same donor. But there are limitations regarding provability post-thaw, poor maturation within the presence of a fetal characteristic even after post-differentiation. However, the culture methods, including techniques and reagents, have improved. And even in the cell handling, which requires a lot of babysitting, can be overcome with advances in automation. So the organoids will have a similar composition to the cell source of orange, with the advantage of keeping the proliferation capacity. It also requires more experienced users, and again, automation can overcome this limitation. So this 3D format has been a high expectation for the future of the precision medicine. And we can go next. So in regenerative medicine, the adult stem cells and iPSC cells are the common cell types of choice. Last year, the International Society of Stem Cells published recommendations to set standards for the stem cell in research, for the use of stem cell in research. And the society recommends that all stem cell lines be subject to a detailed characterization at the acquisition, and having a master cell bank to ensure stock, and everything has to be very well documented for traceability. So the cells should be authenticated, including the cell identity, purity, and sterility, and mutations should be reported and tested periodically for normal karyotypes. Also, the pluripotency for the 3 germ layers should be demonstrated, and they have listed some of the acceptable methods to do it. They encourage transparency when reporting the human stem cells. The cells that are growing in the 3D environment are more stable, though, since it's a more physiological environment, and the organoids has been very well accepted as a functional assay itself. Bioreactors can also be used to grow the cells in 2D or 3D, and the cells can also be cultured with scaffolds or 3D printing, bioprinting, or use the cells a source to build more complex models, such as organoid on a chip, where the cells from multiple tissue can grow in co-culture. And we can go to the next slide. Now, with so many cells growing in co-culture, there are more points to considering. So we are adding variables here. But in any case, the model should be validated to demonstrating the cell phenotype and functionality that recapitulates the in vivo physiology in a context of use. And it's important to check proper differentiation into the major cell types, presence for the targets of interest, when the model is ready to start in the experimental phase, and what is the window for the experimental phase that you have, like what is the longevity of the model. Also determine the reagents compatibility when in co-culture, since when using a media or a matrix for one cell type, it can have a high impact in another cell type. So in summary, what's the more suitable reagent when you're using co-culture? And it is not trivial, so the method should also be reproducible. So not always when you establish methods with one donor, you're going to be working with all, and this can be really problematic. So next slide. So in summary, MPS can support regenerative medicine by providing a highly controlled environment that mimics the human physiology in a context of use. It enables the assessment of tissue response to a drug, toxin, or any other stimuli with a better accuracy than the traditional methods. It can be used to test efficacy and safety of the tissue engineering constructs and optimize the cultural condition for cell growth and differentiation. It provides insights into the mechanism underlying tissue regeneration and disease, and facilitated the development of a personalized medicine by allowing the studies of the patient-specific response to a treatment. And overall, MPS plays a role in advancing regenerative medicine by providing physiological relevant platforms. And we can go on the last one. I think the big takeaway here, it is that with so many technologies commercially available, there is a demand for reliable and reproducible biological resources and protocols that are robust and ideally work across multiple platforms with acceptance criteria. And with so many variables to considering, standards or even start with guidelines are extremely important for the adoption. So that's all for today. Thank you. Thank you for your time.
Great, wonderful. And if you have any questions, you can put those in the chat. I have a few starter questions to get us warmed up, but I'm hoping we can hear from the audience. So jumping in with one starter question, can any group submit regenerative medicine standards to the FDA?
Yeah, so I'll take that one unless you want to, Charlie.
No, it's okay. You can take it.
Yeah, no. The CBER recognition program is looking at the standards developing organizations that fit an ANSI, American National Standards Institute, definition of a consensus standards development organization. And people can play a role in that. And they do need to sort of go through that process of making sure they're a voluntary consensus standard organization. There are other sources like USP that I mentioned earlier and so on that develop standards and so on. But the model that CBER has been looking at for regenerative medicine is around the standards from these sort of community volunteer organizations. There are things like ISO that people can participate in. There's American, what is the, I'm blanking all of a sudden on the other standards organizations, but there are.
ASTM.
Thank you, Josh. That's the one I was looking for. They actually have successfully made some of those standards recognized by FDA. And so I think that's just an important thing to remember, that just about anybody can participate, but that voluntary organization structure that's described in the CBER policy on that is important to keep in mind.
Sounds good. I'm going to jump in with another one, unless there's one from the audience. Why should companies invest time and resources in developing standards and implementing them in their own regulatory processes? Carol, do you want to start off with that?
Yes,
why do we need companies to be working?
I think not only companies, I think companies should invest, participating, but in more like a working group set where we have multiple stakeholders to be engaging and contributing, because there are so many skills that are needed in there, and having multiple set of eyes and contributions in there, it's very helpful. And including the regulatory in their working group.
Yeah, I have a few comments to add on top of that, all those points. I wasn't very explicit about how standards help regulatory review processes and acceptance. And if a sponsor says they are using an assay that's recognized standard, and they say in their application to the FDA, they're following that standard, they can present the results, and they don't need to go through all the validation. They don't need to report all the data, and FDA will accept it on that basis alone. So that's really saves time and money for sponsors of regulatory submissions as well. So yeah, I realize I didn't cover that point, but I think that's the primary motivation on the regulatory side.
Great.
I'll also say that I think if people use some of these standard approaches, there would be increased reproducibility in academic research.
Absolutely.
Yeah.
Another question: how long do standards last once they are recognized?
Yeah, I can.
I don't know the answer. I think Steve can take that.
Steve, how long do they last?
So those voluntary standards organizations say they always include a renewal date or a sunsetting date. They might call them slightly different things, but they're subject to constant revision and renewal. So those dates come from those organizations that put together the standards. And in the future, I think FDA is thinking about including those dates, the expiration dates in that chart.
Yeah.
At least that's been suggested to them. I hope they do. Yeah.
Yeah, no, that's good.
Yeah.
For Carolina, to establish a protocol for human tissue-derived cells, organoids, etc., how many donor sources are used for you to have confidence in the outcome?
I'll say in general, 3 donors would be what we would recommend to check the reproducibility. Sometimes even like from different suppliers as well, because sometimes cells coming from these different suppliers, depending how they were manipulated, also can have an impact in protocol. So ideally, you want to have more than one supplier in hand.
Great.
That was my question. Can I follow up?
Yeah, of course.
In my experience, 3's not enough. So I'm just wondering, and also if you're getting it from different sources, are you using the same collection media? Are you using there are so many different steps along the way to get primary source materials, no matter what they are, whether or not they're bloods or tissues or whatever. And there's such a high variability. Over the years, I don't think that I found that I could use less than 10. Fewer than 10 to
also
feel a high confidence level, as opposed to, yes, you can do it, but you have a high confidence level that you've really worked out the best way.
I think that would be the ideal situation. However, due to the cost and labor of this system it is very difficult to handle multiple donors. That's why, the minimum that you could do would be 3 donors to have the confidence that would be a guide. However, if you're talking, depending on the organ that you're doing, the cell type that you're doing, the disease, you might have to increase that to have any significance.
Yeah, I can comment. Another way to look at it is what's the, desired variability around the measurements that you make,
right?
And how does that fit your purpose for adopting that assay or that standard? And even that, those are very much unanswered questions for the most part. And I think, as Carolina rightly points out, that's, time and effort and resources to establish those things. My personal opinion, based on what I saw at the FDA, is the field would benefit from more of that kind of due diligence and rigor.
And I believe, and that's why it's so important data sharing, right? Because the more users are sharing the data, more comprehension of the outcome of what is needed, we are going to have a better idea and more trust on the system.
Many things that NIST develops go through round-robin multi-lab testing to get real-world experience, and ASTM does a similar thing with some of their standards. So I think a question earlier asked, why companies should do that, and I mentioned the regulatory aspect, but also companies, are required often to develop their own internal standards, reference materials, if you will, and have those sort of qualified for FDA purposes, but they don't necessarily need to share them. So.
And another question, could you give an example of a cryopreservation standard?
I don't have one in mind. I would refer back to the standards recognition document that's available on that website and see. And I haven't looked at that, so it's on my list. But
Jane, are you is that one that you'd like to work on, or you just want more information on standards for cryopreservation?
Sure, I'll work on it. It's actually a really important one, and it crosses the whole ecosystem, all the way from any point where you want to cryopreserve in the whole system.
Yeah.
Yep.
And I don't think anybody's really looked at that for many years.
Yeah.
I mean,
Good.
is it membrane integrity? Is it crystallization internally? Is it the type of crystallization? Is it post-thaw viability or survival or membrane integrity? And what does that all mean? And what would be the best indicators of cell viability post-thaw?
Mhm.
Yeah.
I know there's interest in viability standards and figuring out what viability means and how it relates to anything biological other than dye exclusion. And, that's an important process. I actually don't know if that's sort of one of the major points to consider in that cryopreservation standard I mentioned.
I'm just thinking, but how about mitochondrial function?
Yeah.
Yep.
I did a study with NIST recently that looked at the correlation between trypan blue and other dye exclusion methods and other metabolic assessments, and including things like cell proliferation or ATP production. And the correlation isn't very strong.
Right. You almost
And
have to use more than one.
yeah. And also, I think FDA will be moving towards more of fitness for purpose, fitness for use look at what viability means rather than saying 70% by trypan blue, because they recognize there's a disconnect.
Oh, absolutely. Absolutely. I could think of all kinds of them.
Yep, I know.
I'll throw in another question, and we might end with that unless someone else has a burning one. What are some of the biggest challenges for regenerative medicine standards? Carolina, you want to tackle that one first?
I'm not sure if I'm the best person to do it, but I believe it is the viability of the protocols of viability, how people handling, and usually standards, it is difficult because you do have to be inclusive with multiple stakeholders. I don't know how much more I can talk about it.
Yeah.
So standards across platforms, it's just very difficult.
Yeah.
It depends on the platform. It depends on whether you're talking about one type of cell therapy, whether it's cell gene, a CAR T therapy, or whether it's a cell-based therapy, or whether it's an iPS therapy or an MSC. It depends on cell types as well, platform and cell type. So there's almost a matrix there in which you have to consider whether the standard is appropriate.
Yeah. In my mind, one thought is related to understanding the mechanism of action, how that relates to potency of a proposed cell therapy, and the power of the analytical, tool that's brought to bear. So I mentioned a couple that were in that recognized standard panel, the CFU assay that's related to stemness, and then the osteogenesis one from ASTM. And I think a goal that I think is useful to aim towards is having quantitative bioassays that can distinguish between the same cells made under different conditions, propagated for different amounts of time, or those same cell types from different donors, and getting a feel for heterogeneity and understanding it, being able to measure it.
Yeah.
That's my soapbox about where cell biology needs to catch up to regenerative medicine aspirations.
I have a soapbox too, and that soapbox has to do with the use of monoclonal antibodies that are animal derived to separate your cells out, and FDA getting all cranky about they all haven't been tested for mouse viruses or mouse pathogens or this or that. And it's like, then you go to the vendor and the vendor says, well, of course we haven't done that. And then you have to do, and then the feedback is, oh, well, then you have to test every antibody you've used at every step and since the beginning of God and time. And it's like, okay.
Yeah.
So these kinds of things, these are really, really killers for us, all of us that are doing this kind of work. And and then you have to test the end product, of course, but that's okay. It's an N of 1 as opposed to an N of 20 or something. So,
Right.
you know,
Hmm.
it's,
Mhm.
I mean, and that isn't really always looked at as being a critical component of what you're doing as far as the testing goes.
Yeah.
And then one thing we came across a while back, and we've changed our vendor, but looking at culture bags, and there's certain companies that make bags, and then they use bovine fat, bovine tallow to keep them from sticking together. And then you pick up endotoxin and wonder what the hell it's doing there. Just a point.
Mary Pat, I think you need to help us out with standards.
No, I'm so busy I can't see straight, but I miss this kind of dialogue, you know, as far as it
Yeah.
goes. But FDA is just required, you know, we have 2 clinical studies that we're sponsoring, and so it's been an enlightening moment. But when you go back and you think, well, of course they've tested for viruses, or of course they've tested, but they haven't. And so then it adds a huge burden to those of us who are ready to treat someone who's sick, that we just go, well, and then you change your consent form and say, well, we tested for things, but it still could be something else in there. So your consent form has to include that kind of information for the patient to know that there could be some cooties in there that you didn't pick up.
Yep. It's
Yeah.
a very important, I saw that stumbling block many times. And I do think there's some hope that the agency will start looking at NGS data as opposed to just those viral studies that were the classic ones.
Oh,
But there's progress being made in that field, and ICH has been revised to reflect that, but there aren't many test cases out there yet. So
right. I just thought I'd mention it in case
Yep.
someone's on that path, because it's a little surprises there.
yeah.
All right. Well, with that, let's thank our 2 speakers. Thank you, guys, and thanks our
Thank you. Yeah.
little
Thank
golf
you.
clap.
It was a pleasure.
Thank
And
you.
again, it will be recorded, so we'll share that. And we hope to continue the series. We have some others planned for later on in April, and hope you can sign up and participate in those as well.
Thank you all.
Thank you, everyone.
All right,
Bye.
take
Thank
care.
you.
Thank you. Bye.
Bye.
Bye.